MCAT 2017 Cram, Biochemical and Biological Foundations of Living Systems, Passage 8, HIV and Drug Therapy. Here goes the HIV virus. As you view the reading of the passage, you'll notice some highlighted snippets of text. What I want you to do is garner meaning from these specific selections in order to answer the questions that follow. Good luck and happy reading. Paragraph 1. The human immunodeficiency virus, HIV, is a topic of intensive contemporary research. Hallmarks of the virus include sexual transmission, progressive immune failure, and subsequent deadly infections and cancer. HIV operates by infecting immune system cells, particularly macrophages and T lymphocytes. Paragraph 2. The HIV virus is spherical and contains reverse transcriptase, integrase, and single-stranded RNA. Okay, so here goes your single-stranded RNA, reverse transcriptase, and integrase. It's not depicted here. It is contained by a viral envelope, the viral envelope, which is embedded with proteins from the host cell and a particular glycoprotein referred to as GP120. The GP120 molecules allow the viral cells to adhere to its target receptors located on the exterior of the immune cells, the host cells, okay? Paragraph 3. HIV infects macrophages by binding its GP120 proteins to two proteins located on the macrophages. That's going to be the CD4 receptor, which is important in immune recognition, and the CCR5 co-receptor, okay? The function which is not fully understood beyond HIV. The membranes fuse, they then fuse together, causing the viral RNA to be released into the cell along with some key enzymes, okay? Reverse transcriptase creates HIV DNA, which is then integrated into the host DNA via integrase. So integrase is responsible for incorporating the viral DNA into the host DNA. The host is now referred to as a provirus and continues to replicate the integrated DNA until proteases basically cleave out the viral protein. It is then transcribed into RNA, and the viral components collectively aggregate, and the new HIV cells bud off as a mature virion. This is a complicated as virus. This is crazy. While viruses that undergo a lysogenic or lytic cycle destroy the host cell, HIV does not. For this reason, its life cycle is classified as productive. Paragraph 4. Because GP120 is critical to the primary step of the HIV life cycle, it is a highly conserved protein in otherwise mutation-prone viruses. Scientists have attempted to exploit this conservation in immunotherapy, developing GP120-specific treatments. Maraviroc is a current therapeutic drug that binds the CCR5 receptor, is shown here, blocking the binding of the GP120 to the macrophage cells. And in a clinical study of Maraviroc, over 600 patients were separated into two groups. Group 1 received a placebo drug, and Group 2 received 150 milligrams of Maraviroc twice a day. All right. Table 1, the results of the clinical study, including the decrease in HIV RNA levels. There were no additional adverse events such as illness or death associated with the experimental group. Okay. So I'm not really sure what these adverse events are. If they're not illness, at least there are no deaths. What type of drug is Maraviroc? Is it A, an entry inhibitor, B, a non-nucleoside reverse transcriptase inhibitor, C, a nucleoside reverse transcriptase inhibitor, or D, an integrase inhibitor? You should be able to deduce the answer from the information provided in the passage. I'll give you a moment to think. Press pause and go back and take a look at the passage if you need to. All right. Okay, so although reverse transcription of HIV is prevented with the use of Maraviroc, this is not the stage of HIV's life cycle that Maraviroc acts on. Maraviroc more specifically binds to the CCR5 receptor, which is known to be part of the adhesion fusion process. So with the CCR5 protected, glycoprotein 120 or GP120 can't enter the cell body and form a complex with the CD4 and CCR5 moieties. Therefore, Maraviroc is specifically an entry inhibitor. All right? Okay. Which of the following descriptions of HIV is true? A, HIV is an obligate parasite that undergoes lytic reproduction. B, HIV is a retrovirus that undergoes lytic reproduction. C, HIV is an obligate parasite that undergoes productive reproduction. Or D, HIV is a retrovirus that undergoes lysogenic reproduction. I'll give you a moment to think. You can totally rely on the text for this answer, so if need be, open up a second window to view the reading of the passage and search for this text support to select the correct answer choice. All right. Basically, all retroviruses are obligate parasites. They can't survive without using host cell, nuclear machinery. So HIV creates DNA basically through reverse transcriptase, as depicted here. So it goes from RNA, ssRNA to, I think, shDNA. Okay, that's why it's like reversed, because usually the process flows from DNA to RNA, but this is going backwards, all right? So lytic and lysogenic cycles involve the bursting or destruction of the host cell, whereas HIV doesn't do that. This virion buds off without ever destroying the host cell. It's a very sophisticated retrovirus, okay? It's really serious. So its life cycle is considered productive, all right? HIV does not destroy the host cell and therefore has a productive life cycle. Although that sounds counterintuitive because it does destroy the host on, on a macro scale. Okay, so if Maraviroc targeted the CD4 receptor, as depicted here, what might have been observed in the experimental group? A, there would have been more adverse events. B, there would be fewer subjects with less than 400 copies per milliliter of HIV. C, HIV RNA would remain the same. Or D, there would be more subjects with less than 50 copies a day. I mean, not 50 copies a day, 50 copies per milliliter of HIV. So I'll give you a moment to peruse the table and the passage. Definitely press pause if you need to. Okay. So basically, the CD4 receptor is important because immune cells use it to recognize foreign cells. Okay. If the CD4 receptor is blocked by a drug, the immune cell cannot function to fulfill its function. So, if the immune system cannot function properly, as is the very case with HIV, patients would be more prone to contracting a serious illness or dying, hence more adverse events, okay? So it would enhance rather than, reduce the nature of the virus. Why did scientists choose to target GP120, glycoprotein 120, inhibition in immunotherapy? Is it A, it is involved in the primary step of the HIV life cycle? B, the host cell protects it by conservation? C, an HIV that is resistant to this inhibition is less likely to evolve? Or D, without it, the HIV cells cannot function? I'll give you a moment to think. This one is a little tricky, so be careful. All right. So the passage states that the protein is highly conserved, okay? This means that GP120 is critical to the HIV life cycle. The high conservation indicates that a mutation of GP120 would probably render the HIV to become non-viable. So a mutation that allows HIV to be immune to GP120 inhibition would also likely hinder the HIV's ability to adhere to a host cell. And because a mutated GP120 could not adhere to the host, it could not reproduce. So the HIV strain is not likely to evolve, okay? All right. Where would researchers find the least amount of transmissible HIV? Is it A, amniotic fluid, B, vaginal fluid? I'll leave that up to your imagination. C, sweat, or D, lymph? So I'll give you a brief moment. All right. Okay, so the passage mentions that HIV is a sexually transmitted disease. This indicates that there are transmissible amounts of HIV in the vaginal fluid. So answer choice B is out. HIV also targets immune system cells. Where there is a large amount of immune cells, there will be a higher concentration of HIV. So lymph is out as well. Sweat is involved in temperature regulation and does not contain a significant amount of immune cells. Therefore, transmissible concentrations of HIV would not be found in sweat. Okay? All right.